So today we'll be discussing about cell membranes. You have actually learned about cell membranes in your smaller classes. You know what cell membranes are made of, what their main functions is. So when we talk about cell membranes, again they can be called by various names. Like the most common names you have heard in your smaller classes are plasma membrane, plasma lemma, and the chapter's name itself, that is cell membrane. But today we are going to concentrate more on a broader range of cell membranes. So generally when we talk about cell membranes in the higher classes, we call them biomembranes. What it means is membranes found in biological organisms. So the first thing when we talk about cell membranes, we are going to talk about its structure. So in order to understand the structure of cell membranes, there were three models proposed. The first was proposed by Porter and Mendel. So the first membrane was proposed by Danieli and Davison. So according to them, the lipid membrane consists of lipid bilayers, one on the upper layer and one on the lower layer. The entire lipid layer is covered by a protein coat. Now the thing to remember in this particular model is that the protein coat is not complete. It leaves some holes, and these holes are called polar pores. The function of polar pores is to allow the movement of substances through the membranes. So this model was proposed by Danieli and Davison. Now this model was also not accepted because another model was proposed by Robertson. Again, Robertson is the same thing. Lipid bilayer and covered by protein coat. But according to Robertson, the lipid bilayer is covered continuously by a protein coat. That means there are no such thing as polar pores. So again, this model is also not widely accepted because if this is a continuous protein coat and a lipid layer, that means there will be no movement of substances through the cell membranes. So these two models are not accepted. But the most widely accepted model was fluid mosaic model proposed by Singer and Nicholson. So this is the most widely accepted model if you want to understand the structure of cell membranes. This was proposed in 1972. So according to them, again they take various components from these two membranes. That is, the membrane is made up of lipid bilayers, upper layer, lower layer. But in addition to the lipid bilayers, there are transmembrane proteins. Transmembrane proteins are proteins which span the entire bilayer. In addition to proteins, there are also protein channels. These protein channels allow for the entry of substances into or out of the cell membrane. Then, in addition to the transmembrane channel proteins, you will also find proteins which are present in only a single bilayer. So depending on their position and whether they are in the upper or lower layer, they are known as extrinsic or intrinsic proteins. So this is a lipid bilayer. These are your transmembrane proteins. These are your protein channels. This is the intrinsic protein, and this is the extrinsic protein. Why is it called extrinsic? Because this upper layer is outside the cell, and this one is inner to the cell. So, this transmembrane proteins and channel proteins, what they do is, they help in providing stability to the entire membrane. Why is it called fluid? Is because these proteins and the lipid layers are capable of movement, which I'll discuss later. And why is it called mosaic? Is because the arrangement of the proteins are random, just like a random painting. So hence the name fluid mosaic model. So this is the most widely accepted model of cell membranes. Just remember, protein channels are the ones which allow for the movement of substances through the cell membranes. Now, why is it called fluid? Is because the lipids and the proteins are capable of movement. Now, what type of movement is? They are capable of lateral and flip-flop movement. By lateral, what it means is sideways movement. By flip-flop, it means movement from the upper layer to the lower layer, or vice versa. So, lateral movement is pretty rapid and flip-flop movement is very slow. So, these were also demonstrated experimentally. So, in order to understand this, let's take two mouse cells. One of the cell membranes of the mouse cell I injected with a red dye. Now the injection is done only on the membrane layer there, so the red does not enter the entire cell. The other cell I fuse it with green dye. Then what I do is I fuse both the cells and produce a heterokaryon. Heterokaryon means a mixture of two cells. So what happens is, you're supposed to get only red cells on one side and green cells on the other side. But this does not happen. Rather, what you get is an alternating pattern of red and green color in the cell membrane. So this gives proof of lateral movement. By lateral movement, what it means is sideways movements of the phospholipid layer. So that means if this one was red, if this one was red and this one was green, earlier continuous portion was green, continuous portion was red. But what happens is, due to lateral movement, the red color moves to the other side and the green color in this side. So hence you get alternating patterns of red and green. So this is an example of lateral movement, which is rather rapid movement. Flip-flop movement means movement from the upper layers to the lower layer, which is very rare and also a slower movement. So these were the three models of the cell membrane. Now again, when we talk about the functions, you already know about the functions, like it helps in the movement of substances, and the movements of the molecules always occurs due to either diffusion or osmosis. Now in addition to diffusion and osmosis, cell membranes also facilitate transport with the use of ATP. So if you remember the above or the earlier diagram, transmembrane proteins play a key role in the transport of substances in and out of the membranes. Now, the transmembrane proteins can either be a single protein or a double protein forming a channel. So, let's just take into account this is a single protein. Now, this protein, what it does is, with the use of ATP, it transfers substances in and out of the cell. Now, depending on the movement of the molecules, this movement of molecules can either be called unique ion. Only hydrogen ion is transported. So this is an example of uniport. Symport means two molecules are transported but both in the same direction. For example, hydrogen and glucose, both are being transported in the same direction. So this is an example of symport. Antiport means transport of two molecules but in the opposite direction. That means one molecule is transported to the inner side and the other one is transported to the outer side. A most famous example of this is the sodium-potassium bond, which is a transmembrane protein which utilizes ATP to transfer two molecules of sodium and three molecules of potassium. This is an example of antiport. That means two molecules transported in the opposite direction. Symport means two molecules transported in the same direction. Uniport, only one molecule is transported. So this is what transmembrane proteins and protein channels usually do. They transport substances in and out of the membranes, generally with the help of or use of ATP. So, in addition to osmosis, diffusion and facilitated transport, your cell membranes are also capable of performing phagocytosis and pilocytosis. Phagocytosis means stretching of the cell membranes. For example, let's talk about amoeba. If it encounters a food particle, what it does is the cytoplasm slowly starts to stretch and surround the entire food particle. So after stretching, what it does is the ends of the cell membrane completely touch the food particle. Now what happens is it pinches off. So when it pinches off, a vacuole is created which digests the food particles. So this is an example of phagocytosis. Similarly, pinocytosis is basically dealing with liquid substances, also known as cell drinking. And phagocytosis is basically concerned with solid substances. So these are also the functions of cell membranes. Now what happens is cell membranes are also modified to perform a variety of functions. Basically, the epithelial cells of the cell membranes are provided with certain structures like hair-like cilia or flagella. So one of the most famous or common modification is microvilli, which you find in your small intestines. By microvilli, what it means is small finger-like projections which help in the process of digestion. What it does is it basically increases the surface area so that more food is absorbed. So just compare between these two lines. The upper line is a straight one, the lower line is a curved one. Curved one represents the microvilli. If I were to stretch the microvilli into a straight line, this is the entire length I will get. So if I am comparing with the upper line, that means only this much of length was being utilized to absorb food. But when the microvilli is stretched, I am getting an additional extra length, which also helps in absorption of more food. So obviously, microvilli helps in increased absorption of food. So that's why microvilli are present in small intestines. And microvilli is a modification of cell membrane. Another modification of cell membrane is desmosomes. By desmosomes, what it means is these are small projections of the cell membrane. In addition to the projections of the cell membrane, you also get fine hair-like structures resembling either cilia or flagella. These fine hair or cilia-like structures, what do they do is they help in cellular communication. In your higher classes, you will learn that basically for communication you need nerve cells or nervous system. But in addition to the nerves and the nervous system, even your desmosomes are capable of cellular communication. So your desmosomes are also modifications of the cell membrane. So just remember, when we talk about cell membranes, fluid, mosaic, model. Why is it called fluid? Because it's capable of movements, which can either be lateral or peripheral movement. Why is it called mosaic? Because the protein structure is arranged randomly, just like a painting. Then functions of cell membranes or plasma membrane. Functions, what it means is, most common one is diffusion, osmosis, facilitated transport, and phagocytosis and pinocytosis. And the last thing is modifications. By modifications, it can either be microvilli or desmosomes. So, fluid mosaic model can also be observed experimentally. What are we going to do is, we are going to freeze a cell at minus 196 degrees Celsius. Then what we are going to do is, we are going to use a special knife to cut the cell. So after cutting the cell, so if you cut a cell membrane and separate it into two layers, what you're going to see is you're going to observe proteins and lipid layers. Now the protein layers can be arranged in such a way that they might be present more in the upper layer and the smaller ones in the lower layer. So this experimental proof is also known as freeze fracture technique. Basically what you do is, you freeze the cell at minus 196 degrees and cut it with a special knife. So this is it.